The site-specific incorporation of unnatural amino acids into proteins in live cells was demonstrated by Schultz and coworkers in the early 2000s. This methodology has been expanded upon greatly since that time. These systems are examples of the integration of part and device engineering, as well as the use of device engineering to construct genetic selections. Though we are accustomed to thinking of the genetic code as something universal and static, not all organisms today share the exact same code. For example, mycobacteria use UGA codons to encode tryptophan instead of stop. Additionally, early life is believed to have had a simpler genetic code such as this one at left. This raises the question, what determines the specificity of the genetic code, and can it be expanded to encode more amino acids? The genetic code is determined by enzymatic interactions between the tRNAs, amino acids, and aminoacyl tRNA synthetases in the cell. There are 86 tRNA genes in E. coli, and thus there are more tRNAs than there are codons. Thus, there is not a simple one-to-oneness about the decoding of codons. Additionally, there are around 50 processing and base modification genes that further change the chemical identity of these tRNAs. There are 21 aminoacyl tRNA synthetases in E. coli, which is just one more than the number of proteinogenic amino acids, and there are several deacylation enzymes which can reverse the reaction of the synthetases. Thus, there are a large number of substrates and enzymes that all must interact precisely to retain the specificity of this code. Primarily, this specificity is encoded by the recognition of the synthetases, which must specifically recognize their own tRNAs and amino acids while avoiding all others. Once a tRNA is aminoacylated with an amino acid, little additional editing is done by the downstream enzymes. There are 22 rRNAs, 85 ribosomal proteins, and around 12 elongation and initiation factors associated with translation. But once a tRNA is aminoacylated, that amino acid will get incorporated into protein in response to whatever codon exists in the anticodon of that tRNA. There is some editing by EFTU for mismatched pairings of tRNA and amino acid, but most charged tRNAs are accepted by this molecular machinery. The specificity of translation is around 1 in 10,000, and this is primarily determined by the synthetases. Thus, there exist 20 parallel but orthogonal pathways for aminoacylation in the cell. Leucyl synthetase is exclusively charged with leucine while avoiding similarly structured amino acids. Similarly, all the other synthetases will avoid charging leucine. Similarly, the synthetases must specifically recognize their own tRNAs. The seryl synthetases only charge seryl tRNAs, and so forth. Additionally, there is orthogonality with the recognition of codons. The ribosome will distinguish similar sequenced codons and only employ proper matches. Thus, if one wishes to add an additional amino acid to the code without interfering with normal translation, one must identify a codon that can be encoded, a tRNA that specifically decodes that codon while only being charged by an added synthetase, and that synthetase must only charge the unnatural amino acid. Essentially, this boils down to identifying a pair of tRNA and synthetase that are orthogonal to the others in that organism and specific for an unnatural amino acid. Making tRNAs and synthetases entirely de novo or by computational approaches is well beyond the scope of these tools even today. Thus, it requires protein engineering strategies based on existing enzymes that are close in function to the specification. Thus, other organisms' tRNAs and synthetases were examined for suitability. At the time these studies were performed, this is an ensemble of the knowledge that was known about potential tRNA synthetase pairs. There are 20 proteinogenic amino acids that are common. There are a few others such as selenocysteine and pyrrolysine that occur in some organisms, but much of the natural diversity works with the common 20. Thus, it is highly unlikely that a natural synthetase would accept an unusual unnatural amino acid such as one containing an azide or fluorophore. The altered amino acid specificity is something that must be engineered into the synthetase later. Thus, we must first pick which amino acids to target. The first question is whether there is evidence that a tRNA and synthetase for one of the amino acids will be orthogonal to the recipient organism. Though the biochemical data is sparse, there have been cross-species aminoacylation studies for yeast, rats, some bacteria, and human biomolecules. It must also be the case that the tRNAs for that amino acid can be modified to read another codon in the cell. Of these, the options are nonsense suppression, which means decoding TGA or TAG stop codons, or frame shift suppression, such as decoding 4-base codons and shifting the frame. The challenge here is that many synthetases use the codon itself to recognize their cognate tRNAs, and modification of this region will often render the molecule inactive as a substrate. Since there must be protein engineering to alter the amino acid specificity, there would also need to be a crystal structure. Another consideration would be the cellular concentration of the amino acid. Some, like glutamate, are almost millimolar in the cell, while others are low micromolar. It would be challenging to engineer a synthetase to recognize amino acids similar in structure to glutamate while maintaining specificity for the unnatural amino acid. Finally, the active site needs to be able to accept a new substrate. Mechanistically, some active sites use the backbone to recognize the amino acid, and in such cases, backbone substitutions alone cannot change their specificity. Thus, some amino acids are likely to be workable more than others, and in practice, tyrosine was the best candidate and also the first successful system. Several others, including tryptophan, lysine, and leucine, have since been demonstrated to work as well. The orthogonal synthetase must not recognize any of the native tRNAs in the organism. If it did, the unnatural amino acid would be incorporated into native proteins, and this would be lethal to the cell. It is fairly easy to test this in vitro. There are kits for preparing total tRNA from cells, and the synthetases usually can be expressed recombinantly in E. coli. Also, there are fairly easy radioactive labeling assays for monitoring these reactions. Thus, one can easily ask whether any given synthetase aminoacylates any of the tRNAs of a given cell type. There were several of these studies done in the 1970s which showed that usually prokaryotic tRNAs cross-react with other prokaryotic synthetases, but eukaryotic and archaeal synthetases are sometimes orthogonal to prokaryotic tRNAs. If you examine the phylogenetic trees for tRNA and synthetase genes, you'll see tight clades associated with the different kingdoms. The archaebacteria in particular turn out to be a fruitful area of investigation. Halobacteria are one particular genus of archaebacteria that are growable under normal lab conditions and have tRNAs similar to the sequences of the other archaea. Thus, most of these specificity studies on archaeal synthetases are examined on halobacterial tRNA. The remaining interactions are best studied in vivo using genetic circuits. Because there are so many uncharacterized tRNA modification reactions, it is unrealistic to prepare pure samples of orthogonal tRNAs. One could produce tRNAs using in vitro runoff transcription, but the data from these assays turns out not to be predictive of in vivo behavior, presumably due to the extensive base modifications or other subtle aspects of tRNA recognition by a EFTU or the ribosome. Thus, the specificity of an orthogonal tRNA is best observed in vivo. The easiest codon to recode in E. coli and yeast is the UAG or amber stop codon. If you place a UAG stop codon into a reporter gene, such as an antibiotic resistance marker, it will cause premature termination in a non-functional protein product. If a tRNA exists in the cell that can recode TAG as a sense codon, and this tRNA gets aminoacylated, then the gene's function is restored. The term for such a tRNA is a suppressor tRNA, and these occur through spontaneous mutation, but they can also be caused deliberately by mutation of anticodons to CUA. Several of these selection systems have been developed, allowing researchers to quantify the degree of recoding in the cell. There were conditional positive selectable reporters such as beta-lactamase and chloramphenicol-acetyltransferase, and conditional negative selections such as UP and Barnase. Additionally, there were genetic circuits that could amplify the effect of the suppression and activation of a GFP reporter. The critical requirement for the orthogonal tRNA is that it not be charged by the native synthetases. Thus, in a cell that lacks the orthogonal synthetase, this tRNA should be uncharged and not suppress TAG codons. Phenotypically, then, a cell with one of these reporter genes adjusts the orthogonal tRNA gene should be off, but the addition of the orthogonal synthetase should turn it back on. The reality is that most systems grafted from other organisms do not give clean Boolean results like this. There's usually some gradation where some suppression is observed without the synthetase and some higher level is observed with the synthetase. Thus, it was usually necessary to extensively engineer these orthogonal tRNAs to have them operate well. The strategy employed was a combination of targeted mutagenesis and genetic selection. For most tRNA synthetase pairs, the region of the RNA sequence that are specificity determinants for the synthetase are known. Often this recognition is in the loops in the acceptor stem. By leaving unaltered those sites necessary for recognition by the orthogonal synthetase, while modifying those positions that are recognized by other synthetases, it is possible to find variants that are specifically charged by just the orthogonal synthetase. These tRNA libraries are generated using combinatorial saturation mutagenesis. In this case, the theoretical diversity is 4 to the 11th, or around 4 million variants. That large diversity is screened using genetic selections. First, these tRNAs are subcloned into a plasmid and introduced into cells containing one of the negative selection systems, in this case Barnase. If the tRNA is charged by a native synthetase, the codons get suppressed and the cell is killed, removing that tRNA from the population. The surviving tRNA encoding plasmids are then transferred into cells containing a positive selection system in the orthogonal synthetase. If the synthetase still accepts the tRNA, it will be charged and the cells will survive. The result of this dual selection is enrichment for those tRNAs that are functional for translation in the cell, are tolerated by the synthetase, and are avoided by native synthetases. We have now seen two examples of the integration of part engineering and device engineering. Engineered RNAs have been used to construct an orthogonal suppression system, and a genetic circuit was used to perform the selection. The same approach is used to change the specificity of the synthetase towards the unnatural amino acid. One might expect that this is best done with an unnatural amino acid similar in structure to the native one. However, if it is too similar to the native synthetase will incorporate the unnatural amino acid and this will kill the cell. Indeed, many unnatural amino acids are toxic to cells. To fix that, one would need to alter the specificity of the native synthetases in addition to the orthogonal one. In practice, that is unrealistic today, but it is easy to test for natively accepted amino acids by monitoring their toxicity and eliminating any that are incorporated. Changing the specificity of an enzyme is not a common feat of protein engineering. Though there are many examples of relaxing an enzyme specificity, the specification of this system is that the synthetase exclusively accepts the unnatural substrate. The approach taken to find altered subspecificity variants involves saturation mutagenesis of the active site. The first system this was applied to was a tyrosine tRNA synthetase pair from the Archaebacterium Methanococcus genosci. That synthetase was similar in sequence to the bacterial one, and there was a crystal structure for one of the bacterial variants. In that structure, there is a pocket of residues that recognizes the paraposition of the unnatural amino acid, and these residues were targeted with NNK saturation mutagenesis. Thus, there are 20 possible amino acids at each position in 5 positions, so that's 20 to the 5th variants, which is around 3 million possibilities. These synthetase variants were screened using the same basic dual selection used before to improve the orthogonality of the tRNA. Cells are transformed with the library and a reporter construct containing the orthogonal synthetase and a positive selectable marker, in this case chloramphenicol acetyltransferase, containing the stop codon. If a cell receives a synthetase specific for the unnatural amino acid or the native amino acid, it will survive selection on chloramphenicol. The extreme majority of the surviving synthetases will be specific for the natural amino acids, but rare variants can be selected by a negative selection. Here, the pool of synthetase plasmids that pass the positive selection are introduced into cells containing the orthogonal tRNA and a Barnase gene encoding stop codons. The unnatural amino acid is omitted from the media, and thus the only active synthetases are the ones that still recognize the native amino acid. Those wild-type-like variants will thus kill the cell, leaving behind only those synthetases specific for the unnatural amino acid. As of 2006, 30 different unnatural amino acids had been incorporated using one of these engineered systems, and that list has continued to diversify since then.